Today, I'm going to be speaking about some of my research on milkweeds that I did as a graduate student at Chicago Botanic Garden at Northwestern University. And you already heard my talk title, which you can see on the screen. So here's the outline for what I'm going to be covering in my presentation today. I'm going to begin with just a brief discussion on climate change and its impacts on plants. Then I'm going to move into a little discussion of environmental tolerance ranges and some of the drivers of those. And end with the introduction with a brief introduction to milkweeds, which will be the group of plants that everything I'm going to tell you about today is happening within. The bulk of the talk will be presenting results from various studies that I've done in the field and the lab, looking at milkweed performance across life stages under different climates. And we'll begin with evidence of gene flow in the study taxa. Then we'll move into some lab germination experiments. Then we'll go out in the field to look at a field emergence experiment, and we'll end with a common garden study that was looking at flowering phenology. It's been really awesome to have multiple talks so far that have looked at flowering phenology from herbarium records. And this is a different type of phenology talk because I'll be doing live observations of populations from throughout the Midwest that were put together in one spot to experience the same climate. And I'm going to end with a sum up of all that ground that we're going to cover and some implications for conservation and restoration. So as this is a symposium on the theme of climate change, I hardly need to belabor the point, but I do want to just take a moment to remind ourselves some of what we're talking about when we're talking about climate change and native plants. Global warming is one of the most communicated, I feel like, components of climate change, as you can see here in this graph, showing increases in global average temperature from 1880 to 2019. And whether we like it or not, the planet is warming relatively quickly and with no expectation for warming to cease in the near future. And it's not just warming, as we all know. This warming comes with a suite of other impacts like increased flooding, more cold waves and heat waves, as well as longer droughts and stronger storms. So it's a whole lot of factors that we're throwing at our natural communities, and we're all really wondering how they're going to fare. When I talk about climate change in plants, I really like to show these drawings from Robert Krulwich of NPR, which I think do a really nice job of showing how rising temperatures can stress plants and animals. Just like us, they have temperatures at which they're comfortable and those under which they're not so happy. And the response of plants and all organisms to climate change will be determined in part by an organism's environmental tolerance range or niche, which you can see visualized on the bottom of the slide. Most simply put, this is the optimum condition for an environmental variable like temperature, bounded by upper and lower thresholds beyond which an organism cannot survive or cannot undergo a life stage transition like germination. And what I'm dying to know is what drives these environmental tolerance ranges and those threshold values, and is there any way by which we can predict what organisms are at greatest risk by better understanding the drivers of environmental tolerance ranges. So one thought among many that have been explored is gene flow. Plant dispersal traits dictate pollen and seed movement across the landscape, and that geographic extent across which genes are mixing often, if we have a larger area, it'll encapsulate greater environmental heterogeneity, thereby as the extent of gene flow increases, so may the experienced environmental heterogeneity reflected in the genes that selection then is acting upon. So for example, here we have a dispersal limited violet. It's likely sharing genes among individuals that are experiencing more similar environmental conditions than a wind-dispersed aster species. Of course, there are many other components that go into gene flow, such as breeding system and habitat specificity, but just wanted to provide an example with seed dispersal. So while environmental tolerance range varies among species and likely populations within those species, it also varies across plant life stages, as I alluded to earlier. And while climate change appears unlikely to result in widespread mortality in the near future, very small changes in temperature can have dramatic effects on when plant life stage transitions occur, like germination or flowering, which we've been talking about a lot of flowering today. So I'm glad I get to bring in some early life stages into the conversation. And these changes in timing or phenology can impact plant survival, but also interactions with other members of that ecosystem, and ultimately reproductive success and the continuance of that population or species. Moving into the next part of my talk, we are going to be focusing on milkweeds, as I said, or members of the genus Asclepias. And according to Kew Gardens, there are over 200 species of milkweeds globally. Milkweeds are native to the Americas and Africa, as you can see in green on the map, but they've been introduced to Europe, Asia, and Australia. And in Illinois alone, there are 24 native species of milkweeds shown here in a lovely poster from Illinois DNR. And milkweeds are very well known for their support of specialist herbivores, most notably the monarch butterfly, but they're also a very important nectar source for a lot of pollinators. And I should say that most of these milkweeds in Illinois are pretty common, pretty abundant. The ones in orange and red are the 4 rare species in the state. Today, I'm going to be talking all about common abundant taxa. All right, so to begin, I'm going to present that genetic data that I mentioned for the 2 species. And for these 2 species, swamp milkweed, Asclepias incarnata, and common milkweed, Asclepias syriaca. For their many similarities, like having wind-dispersed seeds, there are a few ways in which they differ that I want to note. Swamp milkweed is primarily self-compatible, while common milkweed is primarily self-incompatible. And they show different degrees of habitat specificity. So with swamp milkweed, as the name would suggest, being partial to wet prairies, shorelines, and other wetlands. And common milkweed really is a true generalist. It's found in very rural habitats. You're probably very, very familiar with this plant, like old fields, roadsides, and forest edges. Based on these differences, I expected that gene flow would be more restricted in swamp milkweed than common milkweed, and that perhaps when we investigated environmental tolerance range, we'd also see greater breadth for common milkweed and smaller breadth, more narrow breadth for swamp. So to test this gene flow hypothesis, we collected leaf tissue and extracted DNA from populations of these 2 taxa across the Midwest. Here you can see the sample populations in black compared to the overall distribution of the 2 species, with swamp milkweed in yellow and common milkweed in green. And we took these leaves back to the lab, extracted DNA, and genotyped them using microsatellite markers. Gene flow can be investigated by looking at genetic differentiation among populations using a Bayesian clustering approach in the software Structure. And that's what you're looking at here. This is a visualization of the outputs of a structure analysis. So each individual column is an individual plant, and then each population is demarcated with black lines. The populations are arranged from north to south, and I'm presenting the highest likelihood scenarios generated by Structure, which were 2 genetic clusters, or a K of 2 for common milkweed on the bottom, and K of 5 for swamp milkweed on the top. For swamp milkweed, we can see that most populations show very high level of admixture, so lots of colors mixing together there. But there are a few populations that stand out with greater assignment to a single cluster, and I've marked those with asterisks. And then for common milkweed on the bottom, we really just see incredibly high levels of gene flow and little to no differentiation among those populations, whether within a collection region or across the entire span of the study, down from Missouri up to Minnesota. And while this isn't quite what I was expecting to see, these results do suggest that greater habitat specificity in swamp milkweed can result in populations establishing in more isolated habitat patches, reducing gene flow and leading to greater genetic differentiation. But it certainly wasn't the case in most of the populations, as you can see. So next, we're going to move into our first lab experiment, and we're going to bring one additional milkweed in the mix. You can see it's added at the top of the table there. This is world milkweed, or Asclepias verticillata. Like common milkweed, it's primarily self-incompatible, but it is a little bit more discerning in its habitat preferences, occurring on dry prairies and open hillsides, as well as some roadsides. And based on this, we would expect gene flow to be more restricted than what we see in common milkweed. So similar to our genetic study in terms of sampling, collections were made in these 3 regions throughout the Midwest, centering on the Twin Cities, Chicago, and St. Louis. You can see that in the map in the upper right. Here I'm showing germination in response to increasing cold stratification length, so simulating overwintering under natural conditions up to a period of 20 weeks. And under warm incubation, which is the dotted line, we can see that common milkweed really benefits the least from longer periods of cold strat as compared to swamp and world milkweeds, suggesting that they may be more sensitive to shorter winters, which we know, as Colette was just talking about, are an expectation under climate change. But results did vary among collection region, and not in a consistent way, as I often find working with this group of plants. We had northern collections showing greater sensitivity for swamp milkweed there in the middle, and southern collections showing greater sensitivity for world milkweed on the far right. In the same study, we also tested the impact of cold stratification temperature. These were all under 10 degrees Celsius. 0 to 10 degrees C is thought to be the range of temperatures under which physiological seed dormancy is broken by cold stratification. So we looked at 1, 5, and 9 degrees Celsius specifically, and we found no significant effect of cold strat temperature, with the exception of that one panel in yellow, this one incubation condition of swamp milkweed. You can see that warmer stratification reduced germination for seed from the north and mid collection regions, but not from the south. Taken together, this in the previous slide, it doesn't seem that warmer winters, at least under this 10 degrees C threshold, are going to be an issue, but shorter winters may be. In a related study, we investigated germination response of common milkweed to predicted future temperatures using a move-along design, where plates were moved from one temperature regime to another to mimic natural conditions, following seed dispersal in fall through the typical timing of germination in the spring. And here each line is an individual population, and each row is a collection region, with the northernmost collection region on the top, Minnesota. And we found that under future warming, more seeds germinated in the fall following dispersal. And this was most dramatic in Minnesota, that top row, where you can see there was no fall germination under the year 2000 scenario, and 50% germination in the fall in simulated 2085. Importantly, there was still variation among populations. Populations are not all going to respond the same. But this is problematic because obviously, depending on the severity of the winter, but in many cases, a seed that germinates in the fall is not going to make it to spring. So this is basically a lost seed. Now we're going to move out into the lab and look at the 2 field studies I mentioned. The first uses a space for time, or space for climate rather, to evaluate plant regeneration from seed under future warming. Here you can see a map of our 3 now familiar collection regions. And I want to call your attention to the 2 blue stars outside of Chicago and outside of St. Louis. Those are our field sites. And on the right is a graph of mean temperature experienced by the populations I collected seed from, and then also the field sites. And you can see that those really nicely cluster. So the climatic conditions of the population experienced match that local field site. And then all together, they make this really nice series of kind of climatic steps for looking at from coolest to warmest. So first, we will look at germination phenology, now out in the wilds, not in an incubator in the lab. And we can start with swamp milkweed on the left. The top row is the results from the northern field site in Illinois, and the bottom row is the southern field site in Missouri. And each of those points represents a fitted mean for an individual population, with the color being the collection region. We can see that although there was some variation across the germination period for swamp milkweed, performance followed a pretty consistent latitudinal pattern where southern sources germinated earlier and to higher proportions, followed by central and northern sources at both sites. Conversely, the common milkweed on the right, northern populations showed greater germination earlier in the spring, but variation among the collection regions essentially disappeared by the end of the germination period. They all caught up to each other and no one ended up really with an advantage. Here we can see performance across all the early life stages I collected data on. So from germination through emergence, survival, and biomass being the final measure of performance that I collected. And it's visualized over the latitude of seed origin on the x-axis. So you can see those clusters of the southern, mid, and northern collection regions. Swamp milkweed is on the top row and common milkweed on the bottom. And the different field sites are shown with different point symbols. You can pretty clearly see that performance for swamp milkweed varied a lot among the collection regions, with kind of these inconsistent latitudinal patterns across life stages. On the other hand, when we look at common milkweed, we see variation between the two field sites, but not among the different populations that were evaluated at each site. And when looking at our final performance measure, biomass, we can see that the differences between sites basically disappear for common milkweed, but they do persist for swamp milkweed, suggesting that swamp milkweed is more sensitive to the difference in climate between these two sites. Our last bit of experimental results comes from a common milkweed common garden at Chicago Botanic Garden. You can see here an aerial view of the common garden's location in relation to the southern part of the Botanic Garden grounds. It's outlined there in white, and it's directly adjacent to our propagation facilities and nearby some evaluation gardens and a reconstructed prairie. Here we were again looking at populations from these three states, Illinois, Missouri, and Minnesota, one population from each. And as an REU student, Amenya Valdez, pictured on the right, collected data on bloom times and pollinator visits. And we also documented a fruit set and some interesting patterns, which I'll show you now. So here is a graph of the number of plants in peak bloom. We found that northern plants, which are growing under warmer conditions, so plants taken from Minnesota are experiencing warming compared to their normal, what the conditions they were adapted for. They bloomed first, followed by Illinois and then Missouri. The horizontal lines on the graph show you the mean day of peak flower for each population for the 2 years over which we collected data. And I think it's really interesting that the pattern persists in terms of the sequence, but there's much greater interannual variation in southern populations than in northern ones, so the distance between those two lines. You may also have noticed that for Minnesota, peak flowering is much shorter and more synchronous, whereas in the southern populations, peak flowering becomes elongated. So it's a much longer period, but with less individuals in peak flower on any given day. And here are some data from Amenya's pollinator observations. So she recorded pollinator visits to each population over time. Population and seed source are synonymous here, so I apologize that there's some inconsistency in the language. Unfortunately, we couldn't do pollinator observations during the peak flowering for Minnesota, just based on the timing of the REU internship, but we can compare visitation to the local and southern populations during peak bloom. And you can see that Missouri in that lightest gray is what we found. The most pollinator visits were happening to the Missouri population, followed by Illinois, and then followed by Minnesota. But again, we were only at the tail end of the flowering period for that population. And this is where the rubber hits the road, right? So at the end of the season, we counted the number of fruits produced by each plant, differentiating between pods or follicles that appeared viable. So in the top part of that picture, you have like a very nice, I would say like full-size green Asclepias follicle. On the bottom, you can see those little brown, scrawny, underdeveloped fruits. I scored those as aborted. And we summed those all up, and we found that this latitudinal pattern that we saw in flowering persisted. So northern plants produced less viable fruit and more aborted fruit than our southern plants. And we suspect that phenology-related pollinator visitation did impact the fruit set that we're seeing, although it's hard to say that definitively given the limitations of this common garden. And this suggests that populations of common milkweed may have diminished reproductive output under warming, given those limitations. Okay, now I will summarize the findings we just walked through. In terms of plant traits as predictors of gene flow, we didn't find quite what we were expecting. Some, but not most, populations of swamp milkweed showed genetic differentiation, while overall, both species show high levels of gene flow within and across collection regions. In the lab germination experiments, we found that the habitat specialist showed greater sensitivity to shorter winters, but no sensitivity to warming winters under 10 degrees Celsius. And critically, the move along study with common milkweed found increased autumn germination under warming, which was much more pronounced in the northern populations. In the field, we observed the habitat specialist swamp milkweed displayed greater climate sensitivity as measured by biomass accumulation between sites, whereas common milkweed seedlings did not differ. And lastly, the common milkweed common garden found northern populations subjected to this space for time warming may experience reduced fruit set, potentially as a result of maladaptive flowering phenology. So these results are interesting for understanding how close related plants will vary in their response to warming, and how responses vary across life stages from germination all the way through to seed set. While none of the plants I discussed today are rare across the landscape, changes in their abundance or phenology and their reproductive output does have ecological implications, including plant-animal interactions. As is not news to anyone, but I guess as this work contributes to, greater attention should be paid and more resources invested into studying and conserving the more climatically sensitive taxa. And as has been hotly debated for rare plants, assisted migration or managed relocation of populations within or beyond their range could be advisable to maintain climatic fit. Most readily, I feel this movement of plant material could occur through intentional seed sourcing for ecological restoration. Seed sourcing could even be designed to increase trait diversity where that was desirable, such as extending the bloom period of a single species to support pollinators or other associated taxa. And overall, I hope you take away from this talk that milkweeds will vary in their response to climate change, and gene flow may explain some of this variation, but it is far from a perfect match, and these drivers require more investigation. So with that, I'd like to make some acknowledgements. All of my co-authors for the various papers I drew from for this talk, including those that are still in revision or preparation. A number of them are out in the world for you to enjoy, and feel free to reach out to me if you can't access any of them. I would also like to thank my graduate institutions and my many funding sources, including the Illinois Native Plant Society, which supported another portion of my milkweed genetic work that wasn't really on theme today. So I didn't talk about it, but it was looking at variation between wild and commercially available milkweed seeds. And lastly, I'd like to thank my field sites for letting me host my experiments there. So that is all I have prepared to share with you all. There is my email address at the bottom of the screen if you want to reach out about anything milkweed related. I'm always excited to talk milkweeds, and I'd be happy to take any questions that you might have.